Hello, my name is Katie Garman, and I'm presenting on behalf of my lab at Duke University and the Blixlager lab at North Carolina State University. Together, our labs have developed an in vivo porcine model of esophageal injury and repair in order to study the response of esophageal submucosal glands after injury to the esophageal epithelium. We developed this model because of literature supporting the hypothesis that esophageal submucosal glands might play a role in repair after formation of an esophageal ulcer, as could occur in erosive esophagitis where the squamous layer is damaged. One of the current theories is that the esophageal submucosal glands are a protected stem cell niche. It's also important to note that traditional rodent models cannot be used to study esophageal submucosal glands because the glands are absent from mouse and rat esophagus. For this reason, we developed the porcine model in collaboration with the Blixlager lab at North Carolina State University. As you can see in these slides comparing the histology of pig and human esophagus, there are several similarities. Both the pig and human esophagus contain submucosal glands. There are ducts within the esophageal submucosal glands, and these ducts wind their way to the lumen of the esophagus. Both the human and porcine esophagus is normally lined with a stratified squamous epithelium. In humans, we perform endoscopic radiofrequency ablation to treat Barrett's esophagus. This technology was initially developed in pigs, and we use the same techniques used to treat human disease in anesthetized pigs in order to create ulcers that would allow us to study how the esophagus repairs. This image is of a normal esophageal submucosal gland under uninjured squamous epithelium. Here we see an ulcer on the right side of the image 3 days after radiofrequency ablation. Then 7 days after radiofrequency ablation, we observed a ductal phenotype in esophageal submucosal glands as indicated by the arrows. These ducts from the esophageal submucosal glands reach to the surface where the squamous epithelium has been ablated and is regenerating. There are islands of what appear to be multi-layered epithelium from these ducts reaching toward the neosquamous epithelium. Next, we used Phospho Histone H3, a marker of M phase, to determine areas of cell proliferation. The top left image is squamous epithelium from a control animal, and the top right is an esophageal submucosal gland from a control animal. Rare phosphohistone H3 cells are present in these uninjured controls. Then in the lower images, 5 days after radiofrequency ablation, adjacent squamous epithelium has an increase in phosphohistone H3 cells in the basal layer of the squamous epithelium, as we would expect. As seen in the lower right image, at 5 days after injury, esophageal submucosal glands also show an increase in proliferation. While less than 0.1% of cells were phosphohistone H3 positive at baseline, this increased to 0.8% of cells in squamous at 5 and 7 days, and 0.7% of cells in esophageal submucosal glands by 7 days. We use several markers to evaluate the ductal changes in esophageal submucosal glands associated with esophageal injury. Cytokeratin 7 was used as a ductal marker that is present in Barrett's esophagus. In this image at 7 days, neosquamous epithelium is forming on the right, and acini, ductal acini, and ducts are all stained for Cytokeratin 7. Note at the arrow how the neosquamous epithelium connects to the esophageal submucosal gland and duct. Overall, ductal acini became much more common in the esophageal submucosal glands after injury, increasing from about 10% in the uninjured state to about 40% at 7 days after injury. This image includes staining for the squamous marker P63 7 days after injury. P63 is present in the basal layer of the squamous epithelium, around the multi-layered ducts, and around a few of the acini. This finding supports the idea that esophageal submucosal glands can promote healing to normal squamous epithelium. In contrast to the P63 squamous marker, CK8, or Cytokeratin 8, is a Barrett's esophagus marker. We found Cytokeratin 8 in the esophageal submucosal glands, but not in squamous epithelium. In this image, Cytokeratin 8, in the control image in the upper left, is only present in small ducts within the normal esophageal submucosal glands, and average scoring was 10% of acini. Then 7 days post-ablation, cytokeratin 8 expression increased in the esophageal submucosal glands and was present in 60% of activated esophageal submucosal glands. Finally, SOX9 is another Barrett's esophagus marker that is normally absent from squamous epithelium. Interestingly, after radiofrequency ablation, we observe SOX9 in the basal squamous layer and in the esophageal submucosal glands. In the glands, SOX9 positive acini increased from about 13% at baseline to about 65% 5 days after ablation. In this image, the esophageal submucosal gland and ducts are seen in continuity with the neosquamous epithelium in the upper right corner of the image. If you're interested in this video and want to read more about our work, the link to our manuscript is provided here at AJPGI. Thank you.